record to the cloud. Okay, and I think we're recording. Did you all get it? I think it notified you, right?
Looks good.
Okay, cool.
Yep.
Great. Okay, thank you so much, Vanessa. I'm super excited to be here. I've never given a Zoom research talk before, so this will be fun. This is also the first time presenting some of this research that will show up eventually in my dissertation, so I'm really excited. And of course, I'm always excited to talk about Mount St. Helens because it's totally beautiful and lovely. Cool. Okay, so yeah, so the title of my talk today is Microbes on the Mountain, specifically plant microbe associations and interactions on the pumice plain of Mount St. Helens. And here is one of my favorite pictures of the mountain, taken from up towards Johnson Ridge Observatory. Cool. Okay, so here's a little bit of, sorry, I'm gonna minimize me really quick. That way I can not stare at my face while I'm talking. Okay, so here's a little bit of a roadmap of what we're going to talk about today. So we're gonna start off with an overview of plant microbe associations, followed by a discussion of primary succession and how it relates specifically to Mount St. Helens. Then we're going to cover some of the research my lab has done on endophytes and Ulmus-Frankia interactions. And finally, we'll go over some of the implications of these results. Cool. Okay, so what are plant microbe associations? This figure here is just kind of meant to demonstrate how plants can harbor symbionts in both above and below ground tissues. Some microbial symbionts include Actinorrhiza. We'll talk about that more in a little bit, but also Rhizobia, Mycorrhizae, and Endophytes. And I like this figure in particular because it kind of shows you, like, you know, this happy plant that has all of its symbionts and like its happy little phyllosphere, and then the sad plant on the right that's kind of eaten up, potentially drought stressed, and just kind of struggling without these microbe colonists. And so please keep in mind too, so I like this figure too for that reason. It shows A, B, C, and D. So plants can have like multiple associations going at the same time. They can also vary spatially within tissues and temporally throughout the growing season, and they can also interact with one another in different ways. So let's take a look at some of these examples. Cool. So the first one I'll talk about here is Rhizobia. And actually a really good example is this cute little Lupinus lepidus that I took on Mount St. Helens. So they're, if you go up there like in the spring, it's just kind of a beautiful blanket of purple. They're really pretty, like early summer really. And so they are in the legume family, so Fabaceae. And members of this family also include like beans and peas. And what's really cool about rhizobial symbioses is that these bacteria form these nodules on the roots of host plants, and they're able to take atmospheric nitrogen and fix it in such a way that the host plants can then use it. And so, let's see here. Okay, sorry, the Zoom thing was popping up in the way. Okay, so, and this is really important because, you know, in places that are nutrient limited, like the pumice plain, but also in agricultural settings, it's really nice to have this like influx of your own like fertilizer that you can make, and it can reduce the need for commercial fertilizers as well. So the next one is Actinorrhiza. And so these symbioses are, let me admit this person, there we go. So these symbioses also make atmospheric nitrogen available to their host plants. They are more limited in that just 8 families make these associations, including Bacterial, which is represented here with a picture of red alder. Oh, sorry, I'm in Zoom. I'm in PowerPoint. Okay, and I have this little cute cartoon diagram that's just meant to show you like where the root nodules form on a red alder seedling. Let me move my mouse. And so Frankia also form nodules, and they allow their host plants to colonize nutrient-poor substrates as well. And we'll focus on this particular symbiosis in a little bit. Okay, so there's also Mycorrhizae. And so there are several different types. I'll just, I chose a picture of chanterelles here, which are an ectomycorrhizae relationship, just because they're, I don't know, abundant right now and recognizable and delicious. So these mycorrhizae are important for obtaining nutrients for host plants, especially phosphorus in exchange for plant-produced carbohydrates. And then finally, my personal favorite, endophytes. And the round picture here, basically just, it's a microscopic picture that's showing this intracellular fungal tissue. So the little blue lines kind of infiltrating the plant cells there. This is showing, that they colonize plant tissues, not just roots. I have so far shown you just root-associated plant microbe associations, but endophytes can actually colonize all types of plant tissues, from leaves to stems to roots. They can also vary temporally and spatially, even at the scale of like a single plant. So the root tissue and leaf tissue could harbor different assemblages at, oops, sorry, meeting controls. Okay, at different times, during the growing season. They are ubiquitous within plants, and they may occur in any tissue, again, and some studies have actually documented that they're able to confer stress tolerance or pest resistance to their host plants. Both bacterial and fungal endophytes have been studied. I will mostly focus on fungal endophytes a bit later. And as an example, I have Rhizobia punctatum. Sorry. So Rhizobia punctatum is an endophyte of big leaf maple. So I have this nice big leaf maple picture here. And then for like scale, this is about the size of a quarter. So towards the end of like summer, early fall, you see these like black tar patches, tar spots appear on your big leaf maple leaves. And these can actually persist after the leaves have fallen into litter, and they can cause changes to decomposition rates. So it's a really neat interaction. So today we're mostly going to focus on just Mycorrhizae and endophytes, and that just pertains to the research that I'm going to share. So cool. All right, so Mycorrhizae and endophytes both contribute to microbial community processes. And I borrowed this diagram from Oak Ridge National Lab because I really think it ties together different components of ecosystem functioning really well. So we'll see here, we can see here that we have our microbial community processes are just one part of this, big interaction here. And so this includes our ecosystem processes like primary productivity and decomposition. You'll hear a lot about decomposition because that's my thing. I'm really interested in it. So decomp pops up a lot. But it also relates to biogeochemical processes like the nitrogen cycle, which here features our lovely nitrogen fixing bacteria, and also the carbon cycle, which just have a little diagram here for that. Okay, so I wrote this schematic because I think it demonstrates the fluxes really well. And we know that plants need nitrogen in order to grow, as demonstrated by this fertilizer that I just pulled off of Amazon. But basically nitrogen availability in turn predicts our primary productivity. And so this picture is just showing that, on the left, the plants are fertilized, and on the right, they're not. And so the amount of organic material produced is then directly tied to the amount of litter that's available for decomp and nutrient turnover. Okay, so we're going to break this down a little bit piece by piece. So to tie in the Frankia here, so Frankia alni is a species that specifically nodulates all most species, so symbiotic with all alders, but we'll talk about different genotypes a little bit later. They can be found throughout the global distribution of alders. Again, they convert atmospheric nitrogen into a usable form. So it's especially useful in places like the Pumice Plain that are nutrient limited and just kind of a harsh place to be. So they're very beneficial to host plant survival and fitness. And then now I have my foliar fungal endophytes. So I guess just a note that like you can see that there's a green and a brown arrow here and just the orange arrow on the previous slide. Frankia do technically contribute to primary production. I have this here because I'm going to show you stress tolerance specifically. So endophytes again are they're ubiquitous in plants, and they can influence both primary productivity and litter decomposition. So they can also influence stress tolerance. And here I have this picture that I took from a paper by Con et al. And basically what's going on in this picture is the top set were endophyte inoculated. So their stems were treated with a solution that contained 10 different endophyte strains that had been previously found to improve stress tolerance in other Salicaceae members. And then we have this mock inoculated group here that was just given like a control solution. And so you can see that the inoculated plants had a much better drought tolerance than control plants because they are at least still a little bit green compared to our sad mock inoculated plants on the bottom there. And then finally, fungal endophytes can also affect ecosystem processes like litter decomposition. And so I'll use again the example of Rhytisma punctatum because it has been shown to inhibit aquatic hyphomycete colonization of litter during in-stream decomposition, which then retards overall decomposition rates. And aquatic hyphomycetes are just referring to the primary decomposers of in-stream litter. And so these are just like aquatic fungi that enter a stream and start to decompose organic matter during the fall. Okay, so that's great. But how does Mount St. Helens factor into all of this? Well, if you've been to the north side of Mount St. Helens, or where Windy Ridge, like there's a little lookout at Windy Ridge or Johnston Ridge Observatory on the north side, this should look a little familiar to you, at least. And I have, there's like this little orange mat here that was, I think it was about 1 meter long in diameter. So just like kind of like scale here, because it's like a curved picture. So, and that map thing was just for, so there's a little drone on it that my undergrad advisor was flying that day, and it was super cool. But basically, this picture, the main takeaway from it is that it's pretty empty. A lot of the vegetation is small and sparse. And so we've kind of touched on this a little bit with like different hints about nutrient poor substrates, but we're going to take a closer look and then tie all that together. Okay, so this is a before and after picture of Mount St. Helens. So before the eruption, you can see that it's this nice majestic mountain with old growth forest, really lovely, like picturesque. And then after the eruption, Mount St. Helens lost quite a bit of elevation. The Spirit Lake rose, and you can see that there's like trees kind of knocked down in this picture here. So these logs are kind of knocked down here. So basically, during the 1980 eruption, the north flank of the volcano just received pumice of like up to 200 meters deep over 20 square kilometers. Forests were completely obliterated. Spirit Lake was filled with these logs, and you can see this mat here. So they're all kind of interconnected here. They're still there today. You can see them from Windy Ridge. It's really cool. But basically, there was a massive landslide. It just devastated the area, and this area is still recovering after 40 years. And now has served as the study site for the projects that I'm going to describe today, along with many other research projects. It's a really cool natural like laboratory, essentially. So this is a really neat animation that I borrowed from NASA, and it shows satellite imagery before, I think it begins at 1979, and it goes until 2017, yeah. And so you can actually see that the log mat down here is moving around, which is really neat. And so while the surrounding ridges, so you can kind of see it's like very empty, these ridges here eventually kind of green up a little bit over time and gets a little bit of vegetation cover. The pumice plain, which is right in here, pretty much remains unchanged by comparison. And so I'm going to start with a textbook diagram of primary succession, which just illustrates, a barren starting point. So bare rock here. And then over time, species colonize the barren substrate, transform it. They contribute organic matter to soil formation and biogeochemical processes. And then as the substrate develops, you can see this organic matter kind of deepening over time. The plant community changes as well. So you see we're moving from lichens to mosses, to grasses, to shrubs, to longer lived trees here. And so barren substrate can result from receding glaciers, as in the case of the classic chronosequence of successional studies in Glacier Bay, Alaska. Also from catastrophic volcanic eruption, as in the case of Mount St. Helens, or also in Hawaii. From sand dunes or floodplains created by receding lake or river waters. And so this is a picture of sand dunes from Lake Michigan, Indiana, and even from anthropogenic land use changes, including abandoned structures like this parking lot in Love Canal, New York. Okay. So across nearly all primary successional ecosystems, plant community succession has been pretty well studied. For example, Lichter, 1998, so this is the figure on the left here, the pair of figures on the left. They determined that transforming dunes, like sand dunes, to forest took about 300 years using a chronosequence on the shores of Lake Michigan. And then Del Moral has published extensively on vegetation development on Mount St. Helens since the eruption. And there's like decades of data for plots that are still maintained and surveyed across the pumice plain. It's really like an incredible amount of information and like papers contributed to the field. So this figure is from 2002. It's from one of Del Moral's papers that discusses how over time poor dispersers had higher colonization than good dispersers in upland study plots. And while a lot is known about succession in plants, very little is understood about successional processes and microbial symbionts of plants, like colonizing plants. So here I have a diagram that's borrowed from it's a really lovely book, Overview of Ecological Responses to the Eruption of Mount St. Helens from 1980 to 2005. It was compiled by 3 key scientists that have studied Mount St. Helens from the beginning. And it highlights a bunch of numerous factors, interactions, and filters that affect eventual community development following a catastrophic disturbance. So, however, glaringly absent here, in my opinion, is the eventual influence on ecosystem processes exerted by interactions between biotic factors and community composition. A paper by Fukami et al. Oops, sorry, let me admit people. Admit. I think I admitted, there we go. Okay. Zoom controls. Okay, so a paper by Fukami et al. 2010 emphasizes that there must be more work to connect ecosystem and community ecology. And he used like little wood discs and looked at inoculating them with different assemblages of fungi, basically, and looking at different like successional trajectories. And so that paper, in addition to the research that will eventually, fill out my dissertation, hopefully will help to fill this gap there. And I like to point out, oops, I'm not in Zoom. There we go. There we go. Okay. I like to point out just this like picture again that I took from Oak Ridge. Excuse me. I think it's just really nice, like ties things together. And it's just a nice way to kind of remember that, the microbial communities are connected to the ecosystem processes, or connected to the biogeochemical processes as well, and they're all kind of interacting in different neat ways. Okay, so tonight I'm going to share with you 2 studies. So the first one is on Frankia diversity, and then the second one we'll talk about is on cultural endophytes. So, all right, let's get started on this one then. So this one is on Frankia diversity in sympatrically occurring red alder. So Ulmus rubra and Sicca alder, Ulmus viridis subspecies sinuata on the pumice plain. Okay, so I'm going to orient us a little bit. So Mount St. Helens is marked by the little red like endpoint thing on the Google map. And it's about 2 hours outside of Portland, Oregon, about 2 hours outside of Olympia, Washington. And the sites I'm about to discuss were established along the boundary trail here on the Johnston Ridge side of the pumice plain. And so took this trail map. I feel like it's kind of helpful to see, and I've got it blown up a little bit here. So you can see Johnston Ridge Observatory is this little blue dot, and then this, like along here, is where the sites were established. So the student that had hiked it kind of hiked down from Boundary Ridge, from the Boundary Trail onto the Truman Trail. And I think there's a couple plots that are like properly on the pumice plain off of the Truman Trail. Okay, cool. So we sampled sympatric red and Sicca alder within 5 plots along a 1.5 kilometer transect. As transects go, this is pretty small scale. So we're looking at variation over a small distance compared to other studies. Sicca alder is dominant on the pumice plain, and also on like the surrounding ridges. So like kind of up here, you can kind of see that. And also in the blowdown zone, and the limiting factor in the plot establishment was the presence of red alder, at least at the time of this collection. So I believe this was also in 2017. And the sampling was conducted by an undergrad in the LSAMP and McNair Scholars Programs. And so LSAMP is the Lewis Stokes Alliance for Minority Participation, which is funded by NSF. So he conducted the sampling and the DNA extractions. And then McNair is a program at PSU that funds first generation low income and underrepresented undergrads who want to pursue PhDs. And they get to do like research projects over the summer. And I just always like to point those out because they're awesome programs. And students really get a great opportunity to be involved in research, writing, and presentations. It's just a really neat experience for everybody involved, I think. So basically, in the 5 plots, 5 nodules per tree were sampled. So he basically just took the roots, and then we eventually separated the nodules from the roots in the lab. But a total of 23 trees were sampled, 11 red alder and 12 sickle alder. These were surface sterilized. Oops. They were surface sterilized using successive washes of bleach and DI water. And then they were extracted using a Sigma extract and amp kit. Both, we did NIF-H and 16S genes. Those were amplified with PCR, and then we sent those for Sanger sequencing. I don't have any cool, exciting pictures of the extraction process itself because it's just a kit. But I included this picture instead of GeoGirls extracting strawberry DNA on the pumice plain last year. And this is another really cool program I like to point out. It's a really awesome outreach program, specifically targeted for middle and high school girls that are interested in STEM, especially geology. And it's run by the awesome Mount St. Helens Institute. So it's just super cool, and I like to let people know about that. There we go. Something's blinking at me, I'm not sure why. Okay, it disappeared. Okay, cool. Let me click back into the PowerPoint. All right, so we also sent samples to OSU, their Cal lab for chemistry, including like nitrogen and phosphorus content. And then sequences were cleaned. Like when they came back, the raw sequences were cleaned in Genius. And then I binned them into OTUs and assigned taxonomy in Mother. I used the Silva. This was for 16S. I used Silva just to confirm that they were Frankia only, and they were. And phylogenetic trees were constructed in RAXML. I then cleaned them up in R, and then R was used for statistical analyses and producing the figures, including cleaning up the phylogenetic tree. Okay, so we have here our little curve. So we found that sampling more trees would have revealed more genotypes, but the Chow-1 calculation placed the expected number of genotypes at 4, which is what we also recovered. And Chow-1 is just a way to estimate like predicted richness based on a supplied abundance matrix. And I have a table here showing the number of genotypes we recovered based on cutoff percentages. So there are 3 cutoff percentages listed here, but all of the rest of the results essentially will only refer to 99.3% cutoff, which was the optimal threshold for NIF-H specifically. We only had a partial 16S sequence. I think the primers that he used were just like the partial 16S sequence. So we did not use those for like any of the trees that you'll see. There's a supplementary tree I can show you then if you really want to see it, but we weren't quite able to make a phylogeny from that. So that's why we used the NIF-H instead. We found a total of 4 genotypes, which is similar to other regional studies of Frankia diversity. A similar study on a different part of the pumice plain found 5 genotypes at the 97% threshold. And I think what's interesting here is how the genotypes were like dispersed across or among the plots. So only 1 genotype was recovered from plots 1 through 4. And then the remaining 3 genotypes were only recovered in plot 5. So you can see that like down here. Okay. Oh, there's my little thing. So plot 5 had the greatest richness. Okay, so I apologize for how tiny this tree is. I could not make it, I couldn't figure out at least how to make it bigger. The printed size is 30 by 30 inches, but I at least thought that maybe this overview, although you can't really read the sample names itself, and it might still be useful to convey the information it's trying to. So this tree is made up of NIF-H sequences from all of our samples in the study, as well as other sessions from NCBI GenBank of similar studies in the region, and they included white alder, red alder, Ulmus viridis, which was a global study, and then also some sequences were just cataloged as Ulmus species, and they were cataloged by Peter Kennedy, I believe. So samples from plots 1 through 4 kind of clustered within this clade here. And then interestingly, samples from plot 5 are sprinkled throughout the 4 clades. Each of the samples that cluster within each of those clades are identical. And so it's really interesting how there's like that variation, here that shows up in the tree as well. So what is going on with plot 5? So I have a principal components analysis here and a heat map of soil chemical features and kind of how they relate to plots. So we can see that in the PCA that the plots are pretty different, with pH strongly associated with plot 1, and a suite of measurements including soil moisture increasing in plot 3. The heat map is showing the relative differences in different features among the plots, with the lighter colors representing lower values and the darker colors representing higher values. We did find that Frankia communities were significantly different among plots, but they were not different among hosts, so we did not find evidence of host specificity. And then we also found that small-scale distance among plots was more influential on community composition than soil chemistry. So let's look at our key takeaways here. We found that there was low diversity, so 4 genotypes at the 99.3% threshold. It's similar, however, to other studies in the region. I think it's important to consider the landscape of our study site, and especially the contrast between it and the sites of other regional studies. So we sampled in relatively young unmanaged mixed species stands along a pretty short transect, less than 1.5 kilometers, which is kind of the complete opposite of other studies, which focused on older managed and or single species stands over much larger distances. There was a study conducted on a different part of the Pumice Plain that's closer to the researcher lot that's towards like the Windy Ridge side of the Pumice Plain. And even that is not directly comparable because it's like the plot types are completely different. Like the plots that they had sampled were more located in like seeps and along a riparian area. And a lot of the plots that we sampled in were drier and not located directly along like a stream. Okay, so we also found no evidence of host specificity in our plots. So while red and sick alder are sympatric, red alder is far less common on the Pumice Plain and also more recently established in sick alder. The small spatial scale also likely played a role, as other studies, again, found evidence of host specificity, unlike greater scales than we did. So more than 1.5 kilometers total in their transect. So here's our pictures. Okay, so finally, we found the highest richness, 3 genotypes in plot 5. So notably, this is the only plot located close to the debris avalanche. The other 4 plots, here's a little picture showing you. So the debris avalanche is this like yellowish, whitish, yellowish color, and then the pyroclastic flow is this pink color. And so, yeah, so the other 4 plots are located on the pyroclastic flow deposits. They're like closer to the lake over here. It is also located in an area that it could be receiving propagules from runoff from the blowdown zone, so trees like located higher up on the ridge, and also from like surrounding thickets, surrounding alder thickets. Unlike the other areas that could have just been, some of them are pretty sparse. They're just kind of alone in their plots. It's just kind of alder and then nothing around them. Okay, so our findings, I guess I'll just read this to you, underscore the critical role that local variation specifically can play in determining microbial community composition, and also emphasize the need to look at dynamics at multiple scales, which we'll look at here in a second. And so at the regional scale, and this is actually looking, so this is Spirit Lake, Mount St. Helens is over here, and this is, you can kind of see the trail kind of winds down this way, and this is where he sampled going towards this direction. And so at the regional scale, differences in how the eruption deposited certain types of substrate contribute to like patch heterogeneity on the Pumice Plain. These deposits and how the eruption affected the surrounding landscape can in turn affect like drainages, and also the existence of refugia where propagules may have survived the blast. And then we also have plot level scale. So regional scale processes can affect differences in plant community composition, but also herbivore presence. So in this case, stem-boring weevils and elk are potential dispersal vectors. And then also edaphic conditions can be affected by these regional processes, and we kind of see that in the heat map of the soil chemical features that we measured. And then finally, we have our host plant scale. And so at this scale, plant chemical defenses can actually directly affect interactions with symbionts, including Frankia. And then at this scale too, I think it's important to consider between patch, the between patch matrix. And so This is basically trying to keep in mind the hospitability um, of the areas between these inhabited patches. So large sections of the pumice plain, as you can kind of see in the background of this picture, are barren, while others are inundated by seeps, which is more similar to this picture here. And if the between patch spaces are inhospitable, it makes it harder for dispersal and successful establishment in other hosts. Okay, so that brings us to the second study here, culturable fungal endophytes of primary successional plants in Mount St. Helens. This is a close-up picture of Diaporthy, which I just think looks really cool and neat. Okay, I'm going to show you this animation again just so we can focus in on a slightly different area of the pumice plain where the sampling of this study took place. So I'll just switch that there. Okay, so most of the vegetation occurs in riparian corridors, specifically down towards the lake along like A and B transects is pretty marshy, and the cover can be pretty dense over like seeps and streams. Much of this area is covered in willow and alder thickets, and actually it's really hard to navigate through there. It's like so dense. Oops, it's not clicking. There it goes. Okay, so this map shows that area in greater detail. We can see there are 4 major streams on the pumice plain. This large braided area here, this is the Willow Creek drainage. It's the largest of the streams. It's spring fed, and it's, I think every time I've been there, even late in the season, it's always flowing, even up by the trail crossing. And then we have, let's see, this is Clear Creek drainage here. You can see it's a lot marshier, except for Willow Lake down here. Clear Creek is marshier, like higher up. They have, let's see, and then, both Camp and Geothermal also have like these separate upper reaches that they eventually join just above the Truman Trail. So you can kind of see the trail winding through here. But basically all these streams drain into Spirit Lake, and the riparian vegetation is dominated by willow. So here's Salix sinensis, here alder here, and then there's a little bit of cottonwood now too. And so those were sampled as well. So these are the 3 riparian species that I focused on in this study. There's also, here we go. So those species also grow at like upland sites where it's a little drier, but conifers are typically more common in those areas. And so the 3 conifers that I sampled were western hemlock, noble fir, and Doug fir. Okay, so field collection was completed in summer 2017. And this map, I know the sites look a little weird, so I'm going to explain a little bit. So I sampled in 20 field sites that were identified by basically walking transects of the pumice plain and identifying where individual trees are and marking what species they were. And then I took that data and clustered it in R to determine like what constituted like a site and was like easily accessible and where I could sample like multiple species at once. And so leaves from these 6 species were sampled over a 3-month field season period. I collected 800 samples from over 200 trees every month from July through September. These things were surface sterilized. They're processed for culture-based study. We used MEA, which is malt extract agar. And if they were not processed for culture-based study, they were then frozen until extraction using a CTAB chloroform method and direct sequencing. And so here's a little picture of what an MEA plate looks like. It's got this lovely golden color. And then this is just a picture of what CTAB extraction looks like. It's on a kit, so it has like, we use 96 well plates, and I just think they make really pretty colors. So I'm always excited to share that. Cool. So the MEA cultures were isolated until azenic. In previous studies we've done, typically there have been multiple things isolated per like leaf section. So these leaves are surface sterilized. I designed like a whole system, a new system where you can sterilize them in bulk using like mesh sleeves that look like what you would put like baseball cards or something into, but they're made of mesh. So you slot the leaf in and then you can surface sterilize them in bulk and let them dry. You section those, you plate them onto the malt extract agar, and then you wait to see kind of what emerges from them. In previous studies, there have been up to 7 things growing out of them, and so it takes quite a bit to like section those out and then isolate them so that way you just have one morphotype per plate. This, I guess you'll see on the next slide, it was a lot easier to do with these plants than metro alder that I've dealt with before. So we use Sanger sequencing again of the ITS region. And again, the raw sequences were clean and genius. And then the clustering and taxonomic assignment was done in Mother. I used the Unite database since they're fungal species. And then I used various packages in R to conduct the statistical analyses and also to generate some of these maps here. Cool. So I originally presented this slide to my committee a few years ago, but I really like the tables that show the discrepancies in how many plates actually had isolates. So you can see here that most of these numbers are pretty low compared to the over 2,000 plates that were made. And so a lot of these they're still sitting in the lab in a box in the corner and there's just nothing ever grew. And it was admittedly a little disappointing at first, but as I started maintaining the cultures, it was a little bit of a relief. So with any culture-based study, sampling more leaves would have revealed more species. However, again, the things that were stored and are extracted for CTAB, they can be used for sequencing on the Illumina MySeq platform. So this will give us a much more complete picture of the entire endophyte community without the biases of like culture-based stuff. So the media that we used would bias against certain species, and there's also a really small percentage of species that can even grow to begin with in culture. So I'm excited for that. That's in the future soon. We found a total of 40 OTUs, so operational taxonomic units. I have here just kind of the distribution among host trees. So you can see here that alder and willow had the most number of OTUs isolated from them. And then you can also see here, this is the class distribution. All but one OTU belonged to Ascomycota. The other one belonged to Basidiomycota. And then the most abundant OTU was assigned to Arthrinium, just Arthrinium species. And it was far and away the most abundant OTU with a frequency of isolation of 45%. And then of those 40 OTUs, we had 18 singletons. So it was quite a few compared to studies of plants in the metropolitan area. Okay, so I have a non-metric multi-dimensional scaling ordination here to show the differences in species composition. We found that the composition differed among hosts, but did not differ over time, so it wasn't affected by time, in the context of the 3 months that were sampled in the study, so July, August, and September of the 2017 field season. So whether or not the trees were deciduous or coniferous also had an effect on community composition. Since deciduous species were more represented in our isolations, about 3/4 of them, I took a closer look at that community composition in that group and found that host species still had a significant effect, but time still only marginally mattered. So you can see that p-value is still hovering 0.08 down here. So over 3/4 of leaves yielded just a single OTU, while the rest had at least 2. There was only one leaf that had more than 2, and it was a cottonwood leaf from site 62 that had the highest species richness of 4. These are abundance graphs, and I think they're really nice because they show kind of how the composition changes over time and between species and also tree types a little more dynamically than NMDS. And you can see here this like lime green color. I'm not great on naming colors, but whatever this color is, this green of OTU1, you can see how it, like the huge influence it has on these compositions. So for example, Nobelfergus, that was all that was isolated from there. And you can see over time, as we approach September, it becomes a smaller and smaller share. Still significantly like large, but a smaller share. All right, so key takeaways from this study. Overall, on the Pumice plain, we observed colonization rates that were considerably lower than those that have been recorded for red alder or even a number of conifers using similar or identical methods. We found OTUs from over 95% of the leaves sampled in the Portland metro area for red alder compared to less than 15% of leaves sampled here. This colonization rate is low, but it is consistent with the colonization rates of needles in Pinus taeta seedlings, which was 14%. However, I would like to note that they are seedlings that were sampled in that study specifically, and we did not check the ages of these plants here, so there's more variation in plant ages here. And again, it's very empty on the Pumice Plain, and I think that that's something to keep in mind, just that like this is a special place and very different from the metro area where we sampled before. Oops, there it is. Okay, so culturable endophyte communities were also, they were dominated by saprotrophs and pathogens, which have implications for litter decomp rates. Saprotrophs can exert priority effects during early stages of decomposition and inhibit colonization by their taxa. So they essentially can have like dibs during litter decomposition, so after senescence and once decomposition commences. We also found high species specificity, which is in line with other studies of endophytes. And I think it's interesting here, like with the low diversity kept in mind. And here's our little graph again. Okay, so we observed no effective time on composition, which is in direct contrast to other studies, including another Pacific Northwest study, actually by my former lab mate, Brett Younginger, Dr. Younginger, which focused on temporal variation in sword fern. There may be multiple reasons for this. For example, plant host age could be confounding this effect, as well as biases in sample isolation. For example, most conifer samples grew nothing. And that could also be because of the way like conifer samples were taken. If we took leaves at the end of the branches, that could be new growth on a conifer that just hadn't been colonized yet compared to a deciduous like alder or willow. And this is, I like this picture because it has my lovely friends, but also the tree that they're sitting next to. This is a sick alder. It is wrapped in bridal mesh for a reason. I used it for a decomposition study that I'm still working on, at least the data part for. But this tree is very small. It's maybe a meter in height, maybe. But I think it shows the scale really well of the trees that like I was working with on the Pumice Plain. They're all pretty small. I think most of them I was taller than. There's only a couple that like were pretty tall, and they were in like the riparian areas. So when we compare the ages of host plants, not knowing what they are compared to like laboratory studies or greenhouse studies that did grow plants from seeds and they do know the ages, it kind of complicates things a little more. But just like showing our cute little tree here. Finally, so given the high frequencies of isolation for both alder and willow and their dominant pioneering presence on the Pumice Plain, these species are likely serving as important reservoirs for plant-associated microbes. And I took this picture towards, this was towards the Windy Ridge side, and you can see that like they're an important part of the landscape. These here are just like really bushy alders. But basically, if you're looking down from Windy Ridge, you can see the spots where willow and alder are establishing. So they are really important parts of the plant community there. So I guess our final takeaway here would be that there are multiple scales at work. So we'll do this again. So environmental filters can affect the host species able to colonize a particular area. On the Pumice Plain, much of the landscape is pretty harsh. And so there's also limited propagules. They need to land in a particular like hospitable patch and be able to survive. And then we also noticed with our host specificity, that kind of complicates the process since a particular host needs to be able to survive in a particular patch, like distribute to and survive there. And then we also are bringing back the between patch matrix idea again that we previously discussed. So there's a pretty infrequent distribution of hosts on the Pumice Plain, as you can see even in this picture. And even if the endophytes are present, they just may not be able to disperse effectively. And that was actually the case of a previous study of Mycorrhizae on the Pumice Plain a little earlier on in its history after the eruption. So, and then finally, so poor dispersal. And then finally, we have priority effects that are at play. And so basically, depending on the identity of the microbe, the colonizing microbe, the order in which it arrives, and subsequent patterns of colonization, that all can be affected. And there was actually a really neat paper that came out by the Busby group at OSU, yeah, Oregon State, that they basically inoculated different orders of colonists, and they found that certain initial microbes can inhibit the colonization of later microbes and change the trajectory of whether or not a plant could be susceptible, like more or less susceptible to certain diseases, which is pretty cool, and obviously has great significance in areas that, you know, on the pumice plain, we found most of the endophytes are pathogenic. So at least in the culturable endophytes. So that's important to consider as well. Okay, so let's distill this a little bit. We covered a couple main things today. So a catastrophic volcanic eruption completely devastated the area, created a new barren landscape, our primary successional model. There are so many propagules that were destroyed during this catastrophic eruption, and now the dispersal ability is more important. And then we also see that there are multiple scales at work on the pumice plain. And I think this is, at least for me, when I think about it, that's probably the most important and applicable thing that this research has at least uncovered for me. And so what are some implications and applications? So each of the studies discussed today ties together the potential for host genotype symbiont and like environment optimization. And this is especially useful for agricultural crops. We also see the importance of considering multiple scales when considering, like just when interpreting results. So often specific models can fail to consider other aspects. Here, for example, we can use endophyte influence on plant productivity and litter chemistry to predict carbon fluxes in new environments undergoing primary succession. These environments will actually be like increasing globally due to climate change. And so we may also be able to apply some of these findings to reforestation and conservation efforts. For example, you know, as we see temperature regimes and precipitation regimes changing, but specifically in the Northwest, we're going to see like more droughts, more frequent and severe droughts. So we could match up drought, like let's say drought tolerant and pest tolerant strains of, or genotypes of alder and Frankia, and be able to preserve canopy cover over streams that are salmon bearing. So has an economic importance as well. Cool, and I'm really hoping, we're hoping that I can continue some of this with some postdoc work, but it's been totally awesome, an amazing treat to work on the mountain. I'm super grateful for it, the opportunity. Just can't beat that view. It's a great place to work. And so I think the last thing I'd just like to include here, I don't know if people are aware of this, and this, I took this from the Cascade Forest Conservancy site. Basically, the Forest Service has been proposing for a couple years now to build a road across the pumice plain. And you can go check this out on their website. They've got lots of great information. But it's such a beautiful place, and it is not a great idea to put this road there. I could talk at length about it, but basically I just want to bring some, I don't know, attention to this fact that the Forest Service is proposing to do this, and it's such a unique and special ecosystem that the idea of putting this road in, even if it's like for a short period of time, like to move drilling equipment, it would just irreparably damage the area. And as somebody that studies in-stream decomposition, it would just completely impact the streams in a negative way. And this is, Mount St. Helens is it's an amazing place because it was protected after the eruption. It's allowed to come back the way, you know, nature is going to progress. And I think that this would be really detrimental. So I just like to point that out in case other people are interested or unaware of that, and just bring a little bit of attention there. And I think that's about it. I've got some cute culture slides, pictures here. Funding was provided by NSF, PSU, and the Puget Sound Mycological Society. And I think that's all I have. So if there are questions, let me See if I can figure out how to open this back up. I minimized the Zoom window.
Thank you, Emily. Folks who are tuned in, if you have questions, you can post them in the chat. I think we're also okay. We haven't been Zoom bombed, so we trust you all. I think people can undo their mutings and turn on their video if you'd like to talk to Emily and ask specific questions.
Do you want me to keep recording or no? Or
Sure, yeah.
okay.
Also, to reply to someone's question, Christy posted the way to get on the email list. I believe one person asked, how do I get on this list? And there's like a self-serve where you just send an email to the address that Christy provided. Okay, thank you so much. That was so great. I posted 2 questions above Christie's, but please write your own questions, folks. My first one was just about site sampling. I recently went to the Pumice Plains and was like, oh my gosh, this would be crazy. I thought of you and was like this, and your transects sound kind of like a lot bigger than some of the transects or plots that I've done in veg work. And I'm just curious, like, did it feel extreme? It looks like an extreme environment to sample in. And
I mean, I think sometimes I have a love-hate relationship with it. I was really sad I couldn't go. I mean, I don't really have, I think I kind of completed my fieldwork last year, but I was just really sad that, this year I wasn't ready for it quite to be over because it's just so lovely being out there. But I think the hardest part is you don't really understand the scale of it. And so I've been like walking around and I was plotting all these trees with the GPS and was just kind of like, oh yeah, it's only like 400 meters. And then I look at it and I have to go through like a gully and like, it's like up there, there's like a big elk standing over here that I'm trying to like avoid. There's like a thicket in the way. So it's just a funny, cool place to be. And I don't know, I really enjoyed every part of it. It's so cool.
nice.
Yeah.
Ben W is writing, how does Frankia diversity differ amongst older soils of similar composition?
I don't know actually. I haven't looked into like the soil ages and I don't know very much about soils in general. I can totally look into it a bit more and get back to you on that. That's a really good question. We haven't actually published that paper yet. The Frankia one that I talked about, it's about to be submitted, but I think that's something that we need to think about. Can I unmute? Oh, hi, John.
Hi. Following up on Ben's question, I had been thinking as you talked about a sort of a theoretical framework for how you would predict how many genotypes of Frankia you would predict. And I think the framework that might, if you haven't checked it out, you might want to, would be a metapopulation genetic theory framework, which makes predictions about how colonization should impact genetic diversity related to the source populations. And I can certainly connect you to a couple of papers on that.
Yeah, that would be awesome.
But hey, it's great to hear where your work is at right now. I really enjoyed it. And I'm really happy that you mentioned the road on the Pumice Plain. And as you know, I'm working pretty hard to oppose that. And it might be helpful for people to hear, like, how would it affect the places that you've sampled and the processes that occur there? I mean, do you think it would have a big impact? I mean, they're talking about a road that would be there for at least 5 years, with dozens of trips across it a day, and that eventually they would pull it out, perhaps, although we actually don't think they would, and supposedly mitigate it. What are your thoughts on that?
I mean, some of my plots will be completely destroyed. Like I couldn't sample there again. I put in a decomposition, in-stream decomposition study all the dates run together now because of COVID, but it was 2019, fall of 2019. We put it in Clear Creek, just north of the trail crossing. So as you move towards Spirit Lake, like past the thickets, and it's like I couldn't replicate the study if I needed to. And Carrie's stuff was right nearby too, and it just, the stream reaches, like we would get so much debris if the road was above it. Plus it's going to impact the soil. It completely changed the course, which I had happened in a pilot study. The bags just were totally left high and dry. Like I came back a couple weeks later and they're high and dry, which just, that was it for those bags. They could introduce like other things from other places. Like I'd be really concerned, like looking at the microbial decomposer community with sequencing, like how much junk would come back in there. And yeah, I mean, I really hope they don't do that, because I think it would just completely destroy the areas that I've sampled, and like the streams would just be, you couldn't really fix them, even if they mitigate it the way that they're talking about it. Okay. Yeah, I missed the, I thought I had a couple more days to submit, and then I went in and I didn't. And so I emailed them, but they wouldn't accept my letter. So yeah, very disappointed. I'm hoping that the notice says good news.
It won't be. I mean, it'll be bad news. The good news is that there is a dedicated group who is ready to fight it.
Awesome.
It includes the Western Environmental Law Center, the Washington Native Plant Society, Cascade Forest Conservancy, Old Broads for Wilderness, and myself and a couple of other researchers and advocates like Susan Saul, who was one of the people responsible for founding the monument. And if people are interested in helping more, the best way probably is to contact the Cascade Forest Conservancy, because they've really been the group that's kind of been the glue holding the other groups together.
Oh, cool. Thank you, Becca.
So Rachel is writing, did you find the chat? Is it helpful for me to read stuff still?
I don't see Rachel.
President SC.
Go ahead and read it.
Sorry. Sorry, I like literally read the name that's like on the thing. Okay, can I generalize about the time span it is taking for the ulna species to colonize the area? How soon were they present at your sites? I'm not sure actually. I think, let me think back to the papers I've read. I know ulnas started showing up, at least in the early surveys in the 90s. And I know just from like personally walking around there that there's more red alder popping up in like the last few years that I've worked there. I started field work there in 2015. And so since then there's like a really like neat patchwork of alders, red alder specifically down by like the floodplains of Willow. And my lab mate is actually Jake Lovelace. He is sampling those and the Frankia there. His, I don't know if his paper is out yet, but he's specializing specifically in like ulmus genotype and Frankia genotype. And he looked at it throughout the Pacific Northwest as well. But he got to sample there and he found some really neat ploidy like results, like some crazy ploidy was going on with the red alders there. So at least in the last couple of years, they've been popping up more, red alder has, but Sitka, since I've been there, I mean, it's always just been there. So I have pretty limited personal experience though. So like John says, yeah, exploded in 2001 and 2003. Yeah, so. Yes. So does ulnus ever occur without Frankia ulni, or can they be grown without it? I did, so I had to find, my advisor asked me to go find some nodules to bring back for a pilot study. And several of the ones that we sampled, they didn't have any nodules at all. We didn't sequence though, like there's nothing to sequence. And I also wasn't involved in like the actual culturing of them, but they didn't have nodules. So yes, they can occur without them. Yeah.
I'll just go ahead and ask this one. I read somewhere, I can't remember exactly what it was, but I think that the Frankia only and the Rhizobium in the Fabaceae, that they were actually, that the thinking was that they were related. It was the related species, or it was like the relationship was established before the species like split apart. So quite a long time ago, because they're not that related to each other. But are they related at all? The fungal
Like
organisms? I can't remember.
Phylogenetically?
Yeah, wish I could remember where I read that, or what I'm thinking of.
I'm not sure how close they are. I know they're both like bacteria, but.
And how about like the Ceanothus? What do you know? What kind? You probably might not know what's.
It's in a different cluster. I like recently delved into the alder. I actually took over that paper from an undergrad, and we recently delved into like the different clusters. I cannot remember which one it is, but a lot of them actually have like promiscuity between hosts. And my lab mate Robin Dove will be looking into that as well. She specifically looked at it in Scotch broom, which was really cool, and she found like host promiscuity there with Rhizobium. And then another like something else that came out. I'm not sure like what Daniel's doing with it or not, but another undergrad had found multiple like bacteria, different types of bacteria colonizing the same nodule of like Frankia nodules from Ulmus. I think they were from Mount St. Helens, but I'm not entirely certain about the project. But yeah, sorry, I feel like my mind just blanked out there. Got like.
Oh, that's all right. That was a pretty vague question, and I can't. I'm trying to find maybe if I can think what that was.
I have one more question, if that's all right.
Hi.
Can you, I assume that when browsers like elk my assumption is that they're dispersing plant-associated microbes as they're brushing the plant. So is that how those are being dispersed? Or is there another way that I'm not thinking of?
I think like what I had looked into was more like droppings rather than like brushing. Like a lot of these, are also just like potentially walking through streams, like moving like fecal matter in that way. So like plant material and fecal matter in that way. And like potentially moving spores in like things like Rhizobia punctatum that overwinter. That's when they produce their spores. And so if they're pathogenic at that, or not pathogenic, but symptomatic at that point, and you can kind of see the tar spots, which I forget what it is. Starts with an M. My lab had sequenced it at one point. They're like little tar spots that show up on the willow leaves, like towards the fall. I haven't seen them on the alder, but just on the willow. You know, when those spores kind of pop up, those specifically are moved like through water, and then also like rain and like things that are potentially moving through it and moving it to different like sites in that way. Yeah, there was actually, I think it was one of John's studies about elk moving vaccinium, like vaccinium, like they were eating vaccinium berries and stuff and dispersing that throughout the area, which was neat. Yeah, and then does that kind of answer something?
Yeah,
sorry, I feel like I get off a little bit.
What I was wondering about, and so I guess it makes me wonder about resiliency of those microbes to be able to withstand passing through the gut of you know, any number of, be it bears or elk or, you know, other consumers.
Yeah, I think that'd be pretty cool to like, I don't know, sequence what's in there and like sequence, you know, the other stuff, but it's like super expensive. If I had my way, I would sequence like all the stuff I find. But I don't think my advisor would be happy with that. It's like I have like enough samples already. But I think it'd be really cool to take a look and see like what their viability is in that way. I just know like I have worked in streams before, and so that's what we looked at was dispersal like through streams, and in that way. Yeah. And I see, oh, decomp studies. So we haven't combusted those yet. And I'm actually still trying to extract the litter from that study because we got shut down in the spring. And so that kind of put a damper on stuff. And yeah, I'm actually pregnant now, so I can't like use the reagents. So it like took longer as well. So I'm still trying to work on that now that I have a respirator and can deal with it that way. But I'm hoping to have some results by the spring, like winter at the earliest. It should be pretty neat. And it was we did a decomp study with Sika alder specifically in 50 micron and 1000 micron like diameter mesh. So window screen mesh, and then like it's like some kind of filter bag. And we're gonna look at the fungal and bacterial communities in those over a 4-week period because it snowed and I had to pull them out of the stream. But I'm super excited for that. So that'll be the 3rd chapter.
Okay. Anybody have any final questions? One new message. Thank you. You're getting congratulations and thank yous.
Oh, this was really fun. Thank you so much.
Thanks so much
Cool
for
to get to
sharing.
talk about it.
Yeah, I think we're coming up on the end of our time. I guess if people want to log out, some of the board can stay on just to touch base is what we did last month, Rachel. And Emily, if you want to stay on just to chat a little bit, so we can say thank you 100 more times.
I need to switch it to you though, right? Like, because am I still who do I switch it
Host. Yeah.
How did the recording go? Did that work? Did it happen?
It says it's still happening. There's a little cloud with a dot in it.
Oh, we can stop recording and see what happens.
Okay. I should do that before I switch it.
Maybe, probably, yeah.
Do I want to stop?